Hello, and welcome back to the Introduction to BioCyc webinar series, Part 5, Proteins and Reactions. I'm Alexander Shearer, speaking for the Bioinformatics Research Group at SRI International, and I'd like to welcome you back to this introductory webinar series. This is the fifth and final part of the Introduction to BioCyc webinar series. So far in this series, we've introduced you to the BioCyc website, its contents, and how to find what you're looking for. Then we started to answer the question, where does E. coli get its arginine, by way of showing you the contents of a typical BioCyc pathway genome database. In doing so, we've seen compound, pathway, gene, and operon pages, as well as the genome browser. Last time, we ended up by showing off the gene reaction schematic, starting with the ARG-D gene page. We're going to begin this final part of the series with a quick review of the schematic and what you can get from it. So here we are, back at the ARG-D gene page. And I'm going to page down, and over here once again we have the gene reaction schematic. Now remember from part 4 that the gene reaction schematic shows the connections between genes, the proteins they code for, and the reactions catalyzed by those proteins. Genes are the purple squares, proteins are the orange circles, and reactions are the blue boxes. For example, here on the gene reaction schematic we can see ARG-D codes for the ARG-D monomer, which becomes part of this bifunctional homodimer that then catalyzes these two reactions. And that's one of the big values of this gene reaction schematic, is seeing this all at a glance. And another big value is pointed out here by the fact that ASTC and ARG-D both connect to this reaction here. You can also see, at a glance again, when a reaction that's catalyzed by your protein of interest is also carried out by other proteins of interest. That could save you a lot of trouble, for example, if you're thinking of knocking out a protein to try to get rid of its enzymatic activity. So now I will go over the ARG-D homodimer and click on it. And this takes us to that protein page. This is an example of a BioCyc style protein page. Now we're looking at a page for a protein complex that consists of a homodimer. In cases like this, there's no separate page for the monomeric subunit. Everything is batched into one page. However, in other cases of heterodimeric complexes or heteromultimeric complexes, you'll actually get the separate pages for the monomers appended onto one big page. So for example, in the case of especially complex heteromeric complexes, such as DNA polymerase III, you'll actually have complexes appended to complexes to make a large comprehensive view of the protein complex you're looking at that lets you, at a glance, subdivide, see which things are carried out by which parts of the complex, and so on. For the moment, we're going to use this enzymatic page as an example of a protein in a BioCyc database, and we'll take a look at what's in a protein page from top to bottom. Up at the very top, we have the name of the protein. If there is more than one enzymatic function, these are often appended to these protein names at the top. For example, since this is a bifunctional protein complex, it has both the activities listed up here, N-succinyl diaminopimylate aminotransferase and acetyl-ornithine transaminase. Next, we have this protein sequence area, which does the exact same thing as the protein sequence from a gene does. So if I click on it, I get the translated protein sequence for this protein. Below this, we have synonyms, again like the gene pages. These are other ways you might search for this. And this is a good synonym set to show off the value of the synonym feature. Because in addition to finding ARG-D by looking for ARG-D, you can see that there are a couple other gene names there, and now protein names, that are from the early research of ARG-D, most likely, DAPC, DTU. And so if you were looking at an old paper and you saw a reference to DTU, you could search it, it would come up as ARG-D, and you can say, ah, I get it. This was determined eventually to be ARG-D, and you don't have to dig your way through the literature to figure that out. Below the synonyms, we have the summary for the protein. Our preference in EcoCyc is to have most of the information about protein function, and by extension gene function, live in the comment area. As I mentioned on the gene page, you could put your functional information there if that were what fit your intuitive model of what's going on in your organism. Again, we're following the assertion citation format. So here we have mention of the initial purification and a reference for that, a later purification, then some information about sequence similarity, and so on and so forth.
Below that, we have a link to the gene that codes for this protein, and because again this is a homodimer, there's just the one gene, but were this a heteromultimeric complex, we'd actually see a listing of the different genes that code for this. Then we have the subunit composition, which tells us it's a pair of ARG-Ds. We have molecular weights, so we have calculated molecular weights for the monomer from sequence, and then we have experimental molecular weights that are culled from the literature. And it's good to be able to put both of these in, because some things, of course, run out very differently on a gel than how they actually would be calculated to run out from the sequence. This may be a function of cleavages. This may be a function of glycosylation if you're looking, say, in a yeast protein. There's a lot of reasons that things might run oddly, and so it's good to know both what the calculated value is, and that's going to matter a lot if you do, say, mass spec, and what the experimental value is, so that you can actually look at a gel and say, oh no, this is okay, this makes sense. Below this, we include the Go terms imported over from the gene page, so you don't have to go over to the gene page to see what Go assignments have been made for that protein's gene. Then as we move down, we have unification links, again like you saw on the gene pages, in this case going to places like RefSeq, ModBase, Swiss Model, Uniprot. In cases where a crystal structure is available, we have PDB links. Then I page down and we have the gene reaction schematic, which we've explained previously.